Namaskar दोस्तों, welcome to channel Toxicology on board. Today in this video we'll be discussing ISHS9 question and answer part 3. So let's directly dive into the question. So question number 20 we are discussing. This section deals with the section 3, that is non-clinical data to support clinical trial design and marketing. So whatever the toxicology data for supporting either phase 1, 2, 3, and for marketing. So those related questions, section 3 related questions we'll discuss here. So question number 20 is start the dose of first administration in humans. Means they're discussing about first in human dose. Reference is made to immune agonist biopharmaceuticals. So here first we need to understand what is immune agonist. We'll discuss that one. Small molecule drugs can also be immune agonist. So that is the reason we need to understand the definition of immune agonist. Can a minimally anticipated biological effect level approach also be used for small molecules? So coming back to first in human dose, no. So before after doing toxicology studies, animal trials, then you will be going for phase 1 study. So what will the first dose that has to be administered to the human? So that is called first in human dose. There are different approaches are there. So we have Noel approach will be there, MABEL approach will be there, based on pharmacodynamic activity, PAD approach will be there. So here the question is related to MABEL approach. Okay. So here generally MABEL approach is applicable for large molecules like biopharmaceuticals or for antibodies. For immunomodulators, which are large molecular weight biologics will be no to that MABEL approach will be applicable. But here there is an exception. Always science will have some or other exceptions will be there. So here in the case of immune agonist, even though it is a bit smaller molecule, still this MABEL approach can be applicable or not. So what is the ISH agency is answering is that if appropriate, exception, a MABEL approach could be used for small molecules using in vivo and in vitro data. So this is an exceptional statement. This approach should be considered if risk factors are derived from the knowledge of. Only when and when this particular MABEL approach can be applicable for immunoagonists, which are small molecules, is number one. If we understand the mode of action, if we understand how that immunoagonist is useful in the case of anti-cancer treatment, if we understand the nature of the target, that is whether the target is a receptor, a protein, then you can understand. Then next, the relevance of the animal. In case of this anti-cancer drugs, no, you have to see whether that particular species in which you are doing the animal trial, whether it is mice, rat, or rabbit, whether is relevant species or not. For example, this particular drug which will go and bind to that receptor, is that receptor available in that particular species or not? Then only that species will be considered as relevant model or relevant species or in vitro models. So relevance of animal or in vitro models. If all these three will be applicable, then for such immune agonist small molecules, MABEL approach can also be used, and that can be used as a if that is first in human dose okay selection. So very quickly we'll go through what is MABEL and what is immune agonist. We'll see. MABEL stands for minimum anticipated biological effect level. Okay, so this is not coming from a toxicological study. It is coming more from a pharmacological. This one, it is a critical metric, means measuring unit to calculate the safe starting dose, that is FIH, for first in human clinical trials, particularly for complex biologics like monoclonal antibodies and immune modulators. As I told you, know, large molecular weight biologics will be no. For that, you will be instead of Noel approach, you will be going for you will be considering MABEL approach. Why this MABEL approach came into so much? Because there was a very important historical incident which took place in around 2006, I guess. They were developing this particular drug. Its code name is TGN 412. So what has happened is that initially with this particular investigational drug TGN 1412, they have done animal trials. They have done in monkey, and they found that there were no adverse effect. The monkey was fine. Then based, they have derived a Noel from that monkey study. They did some fractionation. Generally, Noel will be applying 10 as a fractionation. Then we will converting into HED, that is human equivalent dose. That will be given to the first in human in the case of phase 1 trial. But what has happened in here is that the dose which was selected based on this monkey study, they first started the trial with 6 patients, and all the 6 patients suddenly started showing cytokine storm. As a result, severe proliferation of T cell proliferation took place. So, what was the reason? Then the FDA as well as European agency, European regulatory agency, after doing thorough investigation, they understood that in the monkey there was absence of this particular receptors to which this drug was going and binding. So the drug was not going and binding. That is CD4 receptors will be there, which are involved in immune system. This thing. So monkey was showing fine, but the same drug when it was given to human had those receptors. This drug went and bind to that receptor and started showing that immunological T cell proliferation, those type of toxicity. So please go back and read in detail about this one. From then on only, this MABEL concept is very, very important. So particularly for biologics as well as for immune modulators, this MABEL approach is much safer when compared to Noel approach, the traditional Noel approach. Okay. Some other video, I'll discuss FIH. Various 3 methods are there. How we are defining HED, how we are defining MR, that is maximum recommended safe dose. I'll discuss. Today we'll only stick to question number 20. Next we'll discuss question number 21. Is the question is the use of highest non-severely toxic dose HNSTD in case of anti-cancer drugs. If you look into the objective of this, any 28-day repeat dose toxicology, any chronic repeat dose toxicology study will be, whether it is 3 months or 1 year. The objective is to derive NOAEL. One of the objective, understand the target organ toxicity of that particular investigating chemical. But here in the case of anti-cancer drug, instead of looking for the target organ toxicity, we are trying to derive two point of departure. So if it is rat as a relevant species, in the case of anti-cancer drug, I'm saying we will be looking for STD. STD is severely toxic dose in 10% of the experimental population, whereas HNSTD is highest non-severely toxic dose. This is done in the case of either dog or in the case of monkey. So fractionation of these two doses will be considered as first in human dose. So that is a little bit background about HNSTD. Okay. So is the use of HNSTD to select an appropriate starting dose applicable to biopharmaceuticals? So now the question is, for biopharmaceuticals, can we go for HNSTD or not? So we'll see what is the ICH agency is giving the answer. The HNSTD may be appropriate in determining the starting dose of biopharmaceutical when a drug is not an immune agonist. In the case of previously we have seen, in the case of immune agonist, better to go for MABEL approach. Taking into consideration difference in the binding affinity between animals and humans, as I told you, those receptors should be there. Whatever the relevant species in which you are studying, it has to have those receptors to which the drug will go and bind. Same receptor should be there in the case of human also. That is why it is called relevant species. Pharmacological properties of the biopharmaceutical, including anti-drug conjugate. So this is you can very quickly go through. If you look into a typical toxicology study, as I told you, in the case of dog and monkey, when you are developing anti-cancer drug, you'll be looking not Noel, you will be looking HNSTD. Whereas in the case of rodents, you will be looking STD10. So HNSTD fractionation by 6 if you do, that will be considered as first in human dose. Whereas in the case of rat, STD10 divided by 10, if you consider, that will be considered as your FIH dose for your molecule. Okay. Very quickly we'll go through what is immune agonist and what is anti-drug conjugate with examples. So first of all, what is immune agonist is? It is a therapeutic agent or molecule that will bind to and stimulate the receptors on the immune cells. So what they are doing, either they are binding into the antigen presenting cells or T helper cells, activating pathways that will kickstart or amplify the body immune response. So what is the main thing is immune agonist. They're going to activate your immune system. Okay, that is the keyword that you have to remember. Coming back to what are immune agonist, immune antagonist. Antagonist, what they're going to either block or suppress the checkpoints or whatever the immune pathway will be. Though it is a cascade of reaction will be there. So there will be checkpoint. As in the case of cell cycle, also you have G checkpoint will be there, no, like that. So these antagonists will stop. Example for this immune agonist is currently mode, which is used in the case of basal cell carcinoma. Next, we'll see what is the we'll understand what is ADC. ADC is antibody drug conjugate. So it's a class of a targeted cancer treatment that will combine with the homing ability of monoclonal antibodies with the cell killing power of chemotherapy. Here you can pause the video and go through this particular diagram. You can see here ADC like a vesicle it will form, and it will form in the form of payload or load which will form. So that is why it's a homing ability of monoclonal antibodies with cell killing power of therapy. It is like a bullet. It will enter into the tumor cells and it will blast the cell. So what is happening here is that since there is a targeted precision targeted type of therapy is there, so it will not affect the normal healthy cells. As a result, what happen? The traditional chemotherapy, if you see Taxol derivatives, Taxol, those uh, in addition to the cancer cells, they also affected the normal healthy cells. That's why you will have lot of hair fall, skin epithelial cells which are rapidly dividing. No, body will not be able to differentiate. But if you have a targeted cell type of therapy is there, these ADC they will enter into the tumor cells and they will kill the tumor cells. Okay, by delivering potent toxins directly to the tumor cells while sparing healthy tissue, this anti-drug conjugate improve treatment efficacy, minimize side effects compared to traditional systemic chemotherapy. So example for this is trastuzumab, which is used in the case of breast cancer. So they are only targeting breast cancer cells. Okay. So that was about the question number 21. Now we'll discuss question number 22. Question number 22 states that ICH-S9 states that in cases where available toxicological information does not support a change in clinical schedule. For example, I am developing a certain anti-cancer drug X, okay, and my schedule was I will give weekly once up to, for example, 4 weeks. But now I am going to change my schedule. I want to continue the therapy up to 2 months, and I want to give it twice in a week. So there is change in schedule. Change in schedule means treatment schedule, because tumor might increase or tumor might suppress. So based on that, physician has to keep changing the schedule also. So in such case, what we need to do? An additional toxicology study in a single species usually sufficient. That is what it is saying. Okay. For example, the initial study was done in rat or rabbit or dog or monkey like that. What additional toxicology study should be conducted? If that is a 1 month, whether you have to go for 1 month study or 3 month study, if the 3 month study with the original schedule, so my original schedule for that drug, I have for that particular schedule I have done only 3 month toxicology study, I have already been conducted. So what additional studies I have to do? Agency, ICH agency is stating that generally 1 month study is enough. Okay, if needed, a study of up to 1 month duration should generally be sufficient to support a change in schedule. Even if there is a change in schedule, 1 month study is enough, and to support marketing also. Okay, this study should be available prior to initiation of the clinical trial, before starting phase 1 or phase 2 or phase 3. Even if there is a change in clinical schedule also, one study is enough. That is the answer given by the ICH agency. So please go through this table 1, which is given in ICH-S9 guideline. It will give you very clearly examples. If the clinical schedule, for example, if the particular anti-cancer drug, investigational drug, if you are using once every 3 to 4 weeks, okay. So on Monday your week 1, on Monday you're dosing, again on week 2 on Monday, week 3, week 4. Then what type of toxicology studies are needed? Means single dose toxicology study is enough in a only single administration study is enough. So similarly, you can pause and see if you are giving that particular drug 2 or 3 times in a week. For example, on Monday, then you're giving it on Wednesday or like that. Then what kind of toxicology studies are needed is 2 to 3 times a week, up to 4 weeks is needed. Okay, here schedule is for 1 week, but in animal trials you have to go up to 4 weeks. Okay, like that you have to check yourself what my drug it fits into this particular type of treatment schedule, clinical schedule. Then accordingly you have to plan your toxicology studies. One more key important point I would say that whenever you are reading such tables, please read notes. Here like 1, 2, 3, 4. They are in the form of superscript. Certain times will miss. It will be under footnotes, but lot of information will be given in the footnotes also. So please pause here. You can read it here, or you can go back to the PDF document and read the guideline. Now we'll dive into the next question. Question is question number 23. What general toxicology studies are recommended for continued clinical development including marketing for genotoxic drugs targeting rapidly dividing cells. So some of the anti-cancer drugs are there who are directly affecting the DNA. Okay, so what kind of general toxicology studies are needed for that particular class of drugs we'll see. So for genotoxic drugs targeting rapidly dividing cells. So examples we have, example nucleoside analogs. उसके लिए the example is remdesivir. Remdesivir is adenosine analog. You know, in DNA we have adenine, guanine, cytosine, thymine will be there. So adenosine analog is your remdesivir, which was used in COVID treatment, right? Alkylating agents, cyclophosphamide, and microtubule inhibitors, taxol derivative, that is paclitaxel. So these all things are, see, microtubules, whether it is alkylating agent, they're part of DNA only. They're going to damage your genetic material. So that have anti-proliferating effects. That is, they are rapidly dividing cells like your hair cells or epithelial cells, okay, in the skin, are expected to be consistent across different species. Toxic study in one rodent. What is in the case of rat? How much? 3 month. 3 month rat study is enough for such kind of DNA damaging drugs also are considered sufficient for continued clinical development as well as for registration of that particular drug, okay, to the regulatory body. So key important point that you have to remember is 3 month rat study is enough. Rodent study is enough for DNA damaging type of anti-cancer drugs. Okay. Next question we'll see question number 24. Question number 24 is in the case of ICH-S9 guideline only states that pharmaceuticals planned for use in combination. Now you have to understand whenever a person is undergoing cancer treatment. Now the current scenario, if you see, we are not treating a cancer patient with only single drug. We are treating him with a combination of drugs. We are trying to target the cancer cells from all the directions. Okay, that is the major goal of a physician whenever he is treating the cancer patient. So you will come across this type of combination treatment also in the case of HIV. So whenever you have HIV patient, you're always going for uh, combination treatment of one or two or three drugs, so that the particular person will not develop any resistance. Similarly, in the case of antibiotic therapy or bacterial infections also, combination drugs will be used. So FDC is a very well-known concept in the case of pharmaceutical this thing. So similarly in the case of anti-cancer treatment also, we are targeting with one or two combination drugs. So how to handle, what are kind of toxicology studies are needed for the combination drugs we'll see here in this question number 23. So pharmaceuticals planned for use in combination should be well studied individually in the toxicological evaluation. You have to also understand, for example, there is a drug A, which is an anti-cancer drug, which was initially developed as a monotherapy, and there was certain another drug B, which was also initially developed for monotherapy. But now today we have decided after 2 to 3 years or 4 years, let's combine this A plus B drug. So individually A drug toxicology, battery of toxicology studies are available. Similarly for B drug also, battery of toxicology studies are available. So how are these non-clinical data considered well studied individually in toxicological evaluation? What is that? I will explain you in the below answer. To support a combination study, if needed, when would a dedicated toxicology study be recommended? So we have 2 3 questions in this question. So first we'll understand what is well studied individually toxicological evaluation. As I told, no, A was done in year 2000. After another drug B was done in 2005. Today in 2010 we want to use A plus B together. Already A data is there, B data is there. Okay. So another thing you have to, before going to the answer, I will also say same concept you will come across in the case of mixtures also. Okay, so whenever you're working in there is a certain worker or there is certain person who is commuting through traffic, he will be not exposed to a single chemical. Whenever he is driving, he will be exposed to sulfur dioxide, carbon monoxide, carbon dioxide. So in real life scenario, you will be always exposed to mixture of chemicals. Okay, similarly a person is having a fever, like and you will be taking antibiotic drug, you will be taking a combination of viral also. So like that, a person, for example, a person is anti-hypertensive, means he is having BP as well as diabetes, he will be taking a combination of two drugs. So inside your body, both the chemicals are going at the same time. try to understand. And in the case of cosmetics also, the for their evaluation also, for example, a cosmetic formula is there which is having 1 to 5 components are there. As per European this in cosmetic evaluation, toxicology study on this particular cosmetic is banned. Then you have to qualify each and every component. So A is safe, B is safe, C, D, E. Then A plus B plus C plus D plus E is safe. Like that same concept will come here also. So well, let's dive into the answer which is given by the agency. Well studied individually means a toxicology evaluation sufficient to support clinical studies of the individual pharmaceutical alone. Okay, so we have enough data on drug A, we have enough data on drug B. Okay, if sufficient clinical data, a complete phase 1 or a monotherapy phase within phase 1 are available for drug A as well as drug B. We have phase 1 means we have data in the case of humans. Okay, phase 1 data with the individual pharmaceuticals. Additional non-clinical data may not be warranted. Okay, now there is no need of going for having a combination data. This is the first thing that you have to understand. Next, a rationale to support the combination should be provided. So always you have to give the rationale why you are combining A plus B. Whenever you are combining A plus B, it is going to increase the anti-tumor activity. How it is going to benefit the patient? Is it going to decrease adverse effects to the patient? Whether there is any enhancement in the treatment? Is there any enhancement in the recovery of that cancer patient that you have to see? So rationale to support the combination should be provided, which can include in vitro, in vivo pharmacological data and a literature assessment. So after that also lot of publications will be there when a drug A or drug B was released into the market. So like that literature assessment also you can support. Next, if there are no or very limited human safety data for one of the combination components. For example, I was discussing about drug A and drug B. For drug A, you have enough number of clinical data. For drug B, there is no enough. There is lack of human data is there. A non-clinical pharmacological study of the combination should be considered. So you have to do some pharmacological studies to showing that A plus B, if you are using together, it is going to kill more number of cancer cells. Okay. So in addition to the toxicology studies with the single agent. Okay. Next, for pharmaceuticals that are pharmacologically inactive in a relevant species. For example, we don't have any animal model in which these particular receptors are there. In such case, what you have to do? Assessment of combination can be based on relevant in vitro study because there is no animal model, there is no monkey or dog model in which these receptors are present. Then you have to go for cell lines like kidney cell lines or liver cell lines like that. So you have to do relevant in vitro tests and mechanistic understanding of the target biology. What type of the tumor is there? What type of receptors are where it is going to bind? So target biology understanding is also very important. If the available clinical and non-clinical data are insufficient, we don't have enough human trials, we do not have enough animal trials, establish a safe starting dose of the combination. A dedicated toxicology study may be needed within the combination to establish a safe starting human dose. So it is much more challenging. I feel that there is a confusion in this last statement. Why? Because already above it is saying there is no relevant species. Again, establishing the safe starting dose will be very challenging. But again, they give the answer that if in case there is no relevant species, you can go with in vitro as well as you can go with target biology, that is mechanistic data. Maybe from these things you will be getting this one MABEL approach or PAD, that is called pharmacodynamically active dose can be derived, which can be used as a first in human dose for this particular type of drugs. Okay, so if you see in the market, no example for this FDC is pertuzumab and trastuzumab, which are used in the case of breast cancer. Okay, so one of the thing might be enhancing the activity of the other drug. Okay, so like that you have to go and check in the literature more number of this combination drugs which are used in the case of cancer treatment. Okay, next we'll discuss question number 25. Question number 25 is also something related to FDC only. So ICH-S9 states that data to support a rationale for a combination should be provided prior to starting a clinical study. Before starting phase 1 or phase 2 with A plus B combination, you have to very clearly mention why you are combining A plus B, because these cancer treatment drugs, no, they are very highly cytotoxic. Already treating with one drug is itself is very challenging to the patient. So if you are going to combine two potent drugs, it is going to harm more to the cancer patient. So you have to give clear rationale why you are combining with A plus B what are the data to support a rationale for this combination study? So what kind of literature you can give it to the agency is, the answer given by the ICH agency is, a scientific rationale should be provided to justify a combination clinical study. Okay. Data demonstrating increasing anti-tumor activity by combining pharmaceuticals in the pharmacological studies. So what are those pharmacological studies? Because you are not giving toxicology studies. Here pharmacology is coming into picture. So you have to check for the animal tumor models. You have to develop tumor models. You have to give this combination A plus B, and it has to be able to kill the cancer cells. In vivo, in vitro studies based on mechanistic understanding of this target biology. As I told you, what is your target biology? Is it a receptor? Is it a protein? What is the nature of the target biology? You have to understand. Should be provided to support the rationale of this combination if feasible. Data could be from in-house. You yourself can do these particular studies, or you can get it from a scientific published literature also. That also can be used to support your justification why you are using A plus B. Okay, now we'll see question number 26. So question number 26 is a little bit tricky, something question with respect to enhancer. So here we have, does the ICH-S9 guidance apply to the drug itself having no anti-tumor activity? From the beginning of this guideline only, it very clearly states ICH-S9 guideline is only applicable to those drugs which are used in the cancer treatment, particularly those cancer patients who are at the life threatening stage, means they are on the phase 4 stage of the cancer, such kind of for which there is no treatment available, no? Such type of severely affected cancer patients will be there, no? For that this ICH-S9 guideline is applicable. But here is a tricky, it is saying, is it applicable such as an enhancer? Is this guideline applicable for tumor enhancer, that is such as an enhancer, not tumor enhancer, enhancing the treatment of this another drug, okay, that is intended to be developed as a pharmaceutical combined only with the certain anti-tumor pharmaceutical for the treatment of patient with advanced disease in the late stages of development. If ICH-S9 does apply to this particular enhancer, which non-clinical studies are recommended for first in human, for clinical development, as well as for marketing applications? So first we need to understand what is enhancer also, that we'll discuss. Yes, for this enhancer also, ICH-S9 guideline is applicable. Yes, these pharmaceuticals are within the scope of this ICH-S9 if they are intended to treat cancer. First of all, they should not have any anti-tumor activity, but this enhancer, when it is used with another anti-cancer drug, this is going to it can going to enhance the activity of this particular anti-cancer drug. Data to show the enhancer is non-active should be provided. As such, enhancer should not have any anti-tumor activity. That you have to keep it in mind. Don't get confused. General toxicology studies, safety pharmacology, that is cardiovascular, CNS, as well as neurotoxic, as well as respiratory system. See, reproductive toxicology assessment should be done for this particular combination. Enhancer plus anti-cancer drug. The enhancer alone may have a more limited safety assessment because nobody has not done any battery of toxicology tests on this particular enhancer. So there will be less number of literature safety of this particular enhancer. Either as an arm in the general, you can, what you can do, you can add an extra group, and that extra group should have this enhancer in that one, either as an arm in a general toxicology combination study or standalone general toxicology, or you take a separate toxicology study considering enhancer as the main chemical, and you do the toxicology study for up to 1 month duration, 1 month, that is 28-day toxicology study, keeping enhancer as a main group you can do, or you can hook up to a this combination and keep one group as enhancer group. Genotox studies may be conducted with each pharmaceutical alone or with combination as relevant. Timing of these studies, you have to go back and check table number 1. What kind of will fit into my schedule, you have to see. So before that, you also go through what is an enhancer. It is bit confusing, like previous slide also, because it very clearly says the one which has no anti-cancer activity, it cannot be used. But here again it says it is applicable. These pharmaceuticals enhancer also a part, they are within the scope of this anti ICH-S9 guideline. Here we'll see what is the definition of enhancer to understand better, and we'll see the example also. An enhancer is a substance used in tandem with an anti-cancer drug. Means enhancer with anti plus, I'm saying, to boost its efficacy. For anti-cancer drug, whatever the its treatment efficacy is, it is going to increase more efficacious it is going to become, overcome drug resistance, minimize the required dosage. So initially, if this anti-cancer drug was 100 mg, whenever you are using with enhancer, you can decrease the dose up to 80 mg, right? And also it will help in overcoming the drug resistance. Okay, a well-known example is whenever you're using curcumin along with paclitaxel taxol derivative. Another example we'll see resveratrol with 5-fluorouracil. So often studied in colon cancer. So it is used in the case of colon cancer treatment. Here, whenever you are using this resveratrol, no, it is going to sensitize this chemotherapy. It is going to cause resistant tumor cells to 5-fluorouracil by regulating the survival pathway. So like this, we will come across. This was something related to section number 3. Okay, we have discussed different type of non-clinical toxicology studies that are needed for the case of clinical trials as well as for the case of marketing. We have discussed anti-drug conjugate. We have discussed about immune agonist. We have discussed about uh, uh, we have discussed about enhancer treatment also, how this enhancer is going to help or increase the efficacy of another anti-cancer drug. Okay, so thank you so much. Be curious. For any questions you can always write a mail to aya.priyanka@gmail.com. All the best for your exam.